Protein-protein interactions are involved in virtually all cellular processes. Accurately predicting protein-protein interactions is of importance to biochemistry as well as drug design. Conventional means of designing drugs is the interaction of a small organic inhibitor of an enzyme binding to a narrow hydrophobic binding pocket at the active site. We could expand this narrow definition of a drug target when we expand it to interacting proteins. This could modulate natural or aberrant cellular processes, thereby expand the possibility of developing drugs. In this paper, we've developed a scoring function for the prediction of interacting proteins. So the general process of computational prediction of protein-protein complexes is divided into the sampling stage and the scoring phase. In the sampling stage, we're given the structure of two proteins, and many thousand complexes formed by those two proteins are hypothesized. This pool of hypothesized structures are evaluated in a scoring function to then rank by the likelihood of each structure. Therefore, the goal is to first have at least one correct structure within the docking phase, and then to be able to select the most native-like structure in the scoring phase. To evaluate the scoring functions, we use a set that's called either a benchmark set or a decoy set. Essentially, we're given an experimentally measured structure, which is called the native complex, and we pool that together with the predicted structures, which are called the decoy set. But because we know the native structure, we could then evaluate the quality of that scoring function. Using this decoy set as a training set, we've trained a logistic regression classifier to build a scoring function. To first build a dataset, we vectorize each structure saved in a protein databank file using a force field called KEXA2. Here is a Leonard-Jones potential showing two interacting atoms, and we see there's an equilibrium distance on the x-axis where the potential energy is minimum. We use the interatomic distance between various atom types combined with the parameters stored in a force field to determine the individual pairwise potentials as well as their probabilities. In a typical use case, we would take the product of these pairwise probabilities to get a system-wide probability to use for evaluation of the stability of the system. So we would rank the predicted structures based on those probabilities. In this methodology, rather than taking the product, we save those individual pairwise potentials in a vector form and use that to train a machine learning model to then refine the pairwise potentials given by a generic force field. However, the vectors directly generated using this method would not be suitable for training a machine learning model because we only have one native structure compared to the 1,000 decoy structures. Therefore, the model trained would have a very detailed information about incorrect solutions, but not a lot of information on correct solutions. To handle the heterogeneity in the data, we use the comparison concept between these vectors. We could generate a decoy minus native as well as a native minus decoy. So each decoy would account for a comparison in each direction, and we'll have an equal number of those individual labels. Therefore, our dataset would be completely balanced and suitable for direct training of a machine learning model. Machine learning models are typically criticized for being a black box alternative to well-understood theories. We want to make sure that once we train a model that we understand how that model is working to confirm that the classification task is sound and not based on artifacts or spurious relationships in the dataset. To understand the factors contributing to the classification, we assess the coefficients in the logistic regression classifier that was trained on the entire dataset. Here in the top of the figure, we see the coefficient values sorted in decreasing order, and we see there's a rapid decrease in the value of the coefficients to 0 and a small tail on the negative valued coefficients. This indicates that we have a small subset of features that contributed heavily to the classification task. In the bottom of the figure, we see the density of each type of features categorized by the interaction of different types of residues. Here we see in the positively charged coefficients, we have a high representation of residues with complementary charges, whereas on the negatively valued coefficients, we see like-charged residues highly represented. Also, we have a high representation of small and aromatic interactions in the positively charged coefficient, suggesting shape complementarity is also important in the classification task. For protein ligand docking, important interaction is between polar moieties of the complex, such as hydrogen bonding. In this case, we see the polar-polar residue interaction represented in the negative region of the coefficient distribution, suggesting that it's predictive of the absence of protein-protein interaction. We speculate that implicitly the machine learning model is finding the desolvation term between those two complexes. We benchmarked this scoring function by evaluating it against other scoring functions in use. Here we rank the decoy set and evaluated a metric called the fold enrichment, which represents the elevation and representation of a near native structure compared to arbitrarily choosing it from the entire dataset. Here we see that the logistic regression classifier is able to rank near native structures at a low rank compared to the other benchmark structures. However, it is not sensitive to structures that don't rank at a low rank. In this paper, we presented a new scoring function based on machine learning to predict protein-protein interactions. Please view the original article for the full details of the research.